my name is Alicia Henn. I am Chief Scientific Officer of Biospherics, and joining us here today for our video interview for our Cytocentric blog is Hari Nakshatri, our Cytocentric visionary, who has done amazing work on looking at the importance of physiologically relevant oxygen, most recently in a landmark paper that has come out in Science Advances on how important it is to collect tumor tissue under physiologically relevant oxygen levels because of the difference it has made. And Dr. Nakshatri, I'd like you, if you could, to tell us a little bit about why you wanted to do this research in the first place. What brought you to this place?
Thanks for having me. There are many reasons why we started this. One among them is that a lot of the research is not reproducible. That's one aspect. The main to me is how do we improve clinical translation of laboratory finding? There are many studies that show promising data in preclinical models, both in the lab and in animal work, but they fail in the clinic. So how can we improve our experimental model system to do that? One of my colleagues, Dr. Broxmeyer, had shown that the hematopoietic stem cells, when they are exposed to room oxygen, they undergo rapid differentiation. So if it can happen to hematopoietic stem cells, will it happen to the tumor cells as well? Last 15 years, there has been enormous amount of work done on cancer stem cell. And cancer stem cell field, despite so many papers, it still remains controversial. Some people claim that a certain population of cells, they claim to have cancer stem cell properties, others claim it is not. Could this be due to some experimental methods people are using? So taking all of this together, we thought, let us look into the tumor cells. If we collect the tumor without exposing to room oxygen, always maintain under physiologic oxygen, do they behave differently if the same tumor, a fraction of the same tumor, is collected and processed in room oxygen? So we went through many self-surface markers to figure out what fraction of the cells are mostly affected. That's essentially the paper that we described that. Having done that, since there is enormous data on the cancer stem cells and the targeted drug sensitivity, we thought, does this behavioral change or phenotypic change in cancer stem cells also have an influence on how they are sensitive to the therapeutic targets? The take-home message to me from this paper, forget about all the cancer stem cell phenotype and everything. To me, the important point is, are we measuring the sensitivity of the cancer cells to the drugs under right physiologic conditions? Or measuring them in a room oxygen concentration will alter the data? Is that responsible for some of the drugs which are promising in preclinical studies but failing in the clinic? There are many reasons drugs fail in the clinic. But is it one of the reasons? That's what this study is about, and that's what we are pursuing rigorously now at the moment.
So you're saying that maybe if we are doing drug screening studies at room air, which is where they're all done, that this is the wrong place to be doing it. This is the wrong place to get the right answers for things for inside the body.
I mean, I wouldn't say not everything is wrong, because we had drugs which have gone from the lab to the clinic and they have been successful. We may be missing certain drugs which may be effective, or we are taking some wrong drugs to the clinic. So it's not, from a practical point of view, it is not easy to do every experiment under physiologic oxygen. It is not possible to collect every tissue, particularly in a surgical setting, to collect every tissue for your research in a physiologic oxygen, right? So if you have a drug candidate which you think it is very promising by screening under ambient oxygen, room oxygen, I would suggest repeat those experiments under physiologic oxygen to see whether they maintain the same activity. then proceed with the next step of your preclinical models and then to the clinic. In a way, it is kind of a cost saving to some extent. But by the way I said, limiting this experiment to the way I said, you may be missing some other drugs as well. There may be some drugs which may be working only under physiologic oxygen level. The screening, if you can afford to do it, if you have facility to do it, you can do some screening under physiologic oxygen as well. You may identify a drug which works only under physiologic oxygen, which we have not done it yet, but we hope to do it sometime in the future.
So that's a really interesting idea that we could introduce another step before you go to clinical trials, where you take your findings from room air, from traditional settings, and then repeat that under physiologic oxygen to see whether it still holds true, maybe before you go to animal models. Right, maybe that would save some animals, maybe keep them from having to use so many animals then for these. And maybe, do you think that would make it translate better to humans, being able to use human tissues under physiologic oxygen in vitro? Maybe that would translate better to humans than even using some animal models.
Exactly. So the one of our goal at the moment as we move forward is to collect as many different tumor types as possible under physiologic oxygen, keep a repository of those tumor tissues, and you do all of your drug screening the way you have been doing it, but then take your promising candidate and use these tissues which have never seen room oxygen, then cultured and processed only in physiologic oxygen and test in them, because you transferring the cell lines which are already growing under ambient air to physiologic oxygen, you may not get the real answer. You need to have the tissue which has never seen the room oxygen. So one of the things that we are trying to develop now is come up with a way where we can collect the tissues that are exposed minimum time, as little as possible, to room oxygen, and then place them under physiologic oxygen, cryopreserve them under physiologic oxygen, so that we can do all these critical experiments under that condition. That's our goal.
Wow. That's really ambitious, and but totally doable at the same time.
It is doable. It is going through some regulatory process now, because you need to have the tissue collection procedure to be approved through a number of agencies. So that's where we are in the process of doing it now.
And these are human tumors you're collecting?
Yes, human tumors. In the paper, we collected the ascites fluid, right, from patients who had ovarian cancer or any other cancer which generate the ascites fluid. We did collect them. We did see the difference in physical collection under physioxia versus ambient air. That's the reason is for our motivation to start collecting the human tissue. For example, all the leukemia samples, they can be easily collected under physiologic oxygen and processed under physiologic oxygen for drug screening studies. It doesn't even require additional instrumentation to collect them.
Because it goes into a tube, a vacuum tube, like ascites can too, right?
Yeah, ascites, what we did was we used a syringe, but there is a special cap. As soon as you take it out, you put a special cap and turn it out. All the syringes, everything was previously equilibrated to the physiologic oxygen. They were kept in the physiologic oxygen chamber with this capped needle, and then we brought it out, collected the ascites fluid, and took it over to our physioxia chamber to process it. That's where we found the difference. Yeah,
that seems very straightforward and easy to implement.
Yes. Particularly hematologic malignancies and any of the ascites fluid or pleural effusion fluids, you can easily collect them and process them under physioxia and easily subject those for drug screening. I mean, probably your physioxia chamber has to be modified to have some robotic things to dispense the cells, and it is going to be a bit of investment, no doubt about it.
Do you think that keeping these tissues under controlled oxygen conditions is going to reduce variability that people might see from place to place in their research? Because we touched a little bit on reproducibility and as a translatability issue. Right. Trying to go from from human findings in vitro to human findings in the person is always easier when you have human cells to start with.
Right.
But do you think that controlling the conditions around those might also reduce the variability and speed the whole field forward a little more?
It is, I mean, again, a lot of work, comparison work has to be done before we feel comfortable saying that that is certainly true. I mean, it's not just looking into the phenotype of the cells. We need to look into the genotype of the cells, because once you culture the cells, they undergo mutation. Then you need to figure out whether those mutations and drifting of the cells are similar under room oxygen or physiologic oxygen. So there are multiple steps of evaluation one has to do before conclusively say that, yes, some of the crisis is because of this physiologic oxygen versus ambient oxygen. So my goal here is to open up a new field of thinking, new area of thinking, and I hope others will capture that. And because it's not possible for the one lab to do all of these things, so I'm hoping that it will drive some interest in public to think about doing the experiments in this way.
Well, we certainly see you as a cytocentric visionary for your work in trying to open up this new field of inquiry, and this is so important for biomedical research as a whole. So you think that being able to control the conditions around the tissues and the cells is going to push things forward, help advance things to the clinic faster? Is that right?
Yes.
Okay.
And particularly if one is doing a transgenic model, mouse model for research, for example, they can easily collect their tissues and characterize them under physiologic oxygen. It can be any tumor type, right? Pancreatic cancer, for example, you can easily do it. Ovarian cancer, you can easily, like the way we did with our mammary tumor models, they can all reproduce their work under physiologic oxygen or oxygen level relevant to the pancreas or the ovary, and then see whether they get a same signaling network or not. So that way, using even a transgenic model, we can initially figure out what are the signaling pathways in a particular tumor that is sensitive to oxygen concentration.
And that brings us back to your paper where you were talking about how much that changed the signaling. Can you tell us a little bit about that? What did you find there?
So one of the signaling network, what we found is called the antioxidant response. That's the signaling by NRF2 that we found is different. We also found the Wnt beta-catenin signaling to be different. And there is one more signaling which is studied extensively called YAP. That signaling seems to be different under physioxia versus the ambient air. These are the only three things that we looked into. Then also it looks like the way the cancer cells proliferate under ambient air, they proliferate much faster compared to in the physiologic oxygen that we have been observing all the time. And part of the reason why you see the drugs are more effective in the ambient air is because the cells are proliferating faster. Generally, the cells that proliferate fast are more sensitive to the growth inhibition by drug. So that way, we may be misinterpreting the data of drug sensitivity in that manner as well. Right. So you lose the
predictive value.
Yes.
Of the assay if it's done under the room air oxygen.
Right. Yeah.
I see.
So another aspect we found is the splicing machinery. Alternative splicing machinery seems to be very sensitive to oxygen. We don't know which among the multiple proteins involved in them. So we do think that the splicing is going to be completely affected. So that way the proteome may be completely different. If you just simply look at the protein, you may not see a difference, but there are exons, few exons or few introns may be retained or spliced out based on the ambient or oxygen or the physiologic oxygen. And there was recently a paper, I think it's in glioblastoma, where they found that a gene become oncogene only through alternative splicing without having any mutations. So the alternative splicing machinery can convert a gene to an oncogene. So that way we may be going in a completely different direction. Does this alternative splicing machinery work the same way in physiaxia versus ambient air? So that can have an impact on the signaling network. We touched upon that a little bit in the paper, but we didn't go deep into that.
What other points from the paper do you want people to remember?
I mean, I touched on the signaling pathway, the drug sensitivity, those are the major things. And also looking at how do you characterize the cancer stem cells. Part of the controversy in the field is because we don't have any good functional marker. And are these functional markers themselves are altered because of the oxygen concentration? So there are 3 different fields where our paper may have an impact. One is signaling network, next is drug sensitivity, third is cancer stem cells.
And how room air oxygen may be adding artifact.
Right, exactly. So,
which is going to give us, the wrong answers. So right. This is tremendously important. Right. What kind of impact do you see this having on the rest of the field?
So my hope is that the field will recognize it, and again, the key experiments where you think you are doing a paradigm shift, probably it's better to do it under 2 conditions to make sure that it is really changing.
Do you think this is something that journals and funders should be aware of?
It will take a while because it is not something it can be done right away. Now, it will take a while for funders and journals to require this because the entire infrastructure has to change in an institution. You have to set up a physiaxia BL2 facilities for the cell culture. And all the tissues you need to collect in this manner. It will be years then before these things can become routine. But it's a time to start thinking in that direction.
Definitely. And so could you see something like a core facility set up in an institution for repeating these experiments before they go to publication?
Yeah, that's what I expect will be the next step. Is that it used to be one person's small corner. They did few experiments in hypoxia. That concept is going to change to where an institution will have, in our case, we are already having one, where it is a hypoxia, physiaxia core facility, where people can go in and do their experiments. Right, so that's the easiest or the quicker way to transition to this method.
So do you think if it's a case of like, if we build it, they will come? Do you think that if it's available, that people will use it?
Yeah, exactly. Change is hard.
Yeah, it's really hard. We've really struggled to kind of tell people that they need to improve what they do because sometimes they're not always very receptive to that. Yeah. They, especially if they have a lot of publications and a lot of work built on doing things in room air, it is very difficult to take that investment and say, I need to do things differently. And maybe I need to repeat some of my old studies under physiologic oxygen. That's kind of a risky thing. Yeah. If you have a body of data built up, like what's the best way we can convince people that they need to take that risk?
Right. Again, I mean, as you pointed out, eventually it is the journals and the funders who have to come up with that in a policy, right? But I don't think any one of them is going to implement it right away because it's not practical. So
it's not practical.
No, it's not practical. And what I do expect is the pharmaceutical industries to adapt to this if they want to accelerate their drug discovery and make their drugs more efficient. Instead of academia, it may be a slow process, but I hope these big pharmas where they do all the screening, if they can adapt to this, I think that's the initial step, I suspect.
Mm-hmm. And that will reduce the number of bad drugs that make it to the clinic. That will reduce the number of animals used for drugs that won't work.
Yeah.
But it may not always catch the ones that would work under hypoxia unless you did those initial studies under the correct, I shouldn't say under hypoxia, I should say under physioxia.
Physioxia, yeah.
Because you need to do those initial studies under the right oxygen level in order to get the broadest screenings through in the right conditions.
Right.
So if people are taking skeletal muscle out and doing it in room air oxygen, which is 20% or 21%, in an incubator it might be 18.6% is the highest it could be, because you have the CO2, you have the humidity in there, which you keep it down a little bit more. But all of that is far above anything that skeletal muscle would see inside the body. And so that would skew all those findings.
Right, exactly. Yeah.
Okay, very interesting. So how do we convince people that other people working in skeletal muscle that they need to do it at 3.8% oxygen?
Yeah, I mean, hopefully we will be doing it once I get my grant renewed. Right, I'm working. So that's what I'm proposing. I'm going to compare it in both the physiologic oxygen and room oxygen, the skeletal muscle work we are doing in the lab. So
very cool. Very cool. So are there other things that you want people to know about your work?
I mean, the one other aspect I mentioned is the splicing machinery, where the proteome of the cells can change based on the oxygen. And the splicing was something we didn't give too much attention all these years. But I think the deregulated splicing as a pathology has a pathologic implications is something it's emerging now. Some companies are starting to develop drugs on them. These are targeting the splicing. Again, those drugs also have to be tested under physiologic conditions to figure out whether it works the same way. From the scientific aspect, we need to still try to figure out how many among the 30,000 odd proteins in the body, in a cell, are sensitive to the oxygen level. Their activity is affected by the oxygen level. We only focused on the HIF1 regulated genes or proteins that are made. That is not the story at all. And that's one of the stories of oxygen sensing mechanism in the body. There are many more that are already emerged and many more will come. So that's where the focus has to be.
Wow, this is so exciting. What I'd really like to do is come back and talk to you again, in another year or two, and see how things are going then, because your work is so important to fixing what's wrong with the way we handle cells these days, and pushing forward the right technologies that you need is something that we want to help with. So that you're not burdened with room air, and this artifact that goes into every assay from the room air to improve reproducibility, to improve translatability, and get the right drugs to the patients that so desperately need them.
Yeah, exactly.
Well, thank you so much for talking with us. We really appreciate you taking the time. We know you're a busy guy, and we want to again say that we really appreciate what you're contributing to the research community, and this direction that you have taken, we think can really change the world.
Thank you very much. I appreciate it.
Thank you.
Bye-bye.